Okay, I'm hearing the church bells ring, so I think we can start sharp at 8. Welcome everyone to this week's webinar of Applying Ethology, with a topic that is not only of interest for people that work on animal behavior and cognition and animal welfare, but I think applies to a lot of other researchers as well. Before we're going to start, just two notions or two notes. Please, everybody take care that audio and video is switched off so we can provide the best quality of the presentation. And if you have any questions for the speaker or on specific slides, just drop them in the Zoom chat function. So we can, even if it's an urgent question, we can try to ask it right now, or we can ask all the questions at the end of the presentation. I'm still hearing some voices from some other people.
Can you go? Can you put that on there?
Okay. Okay. Our today's speaker is Ben Farrar from University of Cambridge. He's interested, or specifically interested, among other things, on open science practices, and publication methods in animal cognitive research and animal welfare, which is, I guess, what will be covered in the talk today. He will talk about reliability, credibility, and open science in animal cognition and animal welfare science. So I'm very happy to listening and seeing his talk right now. So the stage is yours, Ben.
Okay, brilliant. And thanks for everyone coming out, whether it's evening or not, for where you are. Yeah, as I said, as Christine said, sorry, I'm going to be talking a bit about reliability, credibility, and open science, particularly looking at it in animal behavior, animal cognition, animal welfare research. Yeah, so what I'm going to talk about is I'm going to split it roughly into 4 sections, about 5 minutes each, so going through quite quickly and then having lots of time at the end for a bit of a chat about it. So I'm going to go through a bit about what is open science. It's a word that encompasses a lot of different things. You might hear of it in sort of open source software, all the way through to reproducible science and the reliability of science. And then I'm going to focus in a bit on the replication crisis, which has unfolded over several scientific disciplines over the last sort of 10 years, 20 years in some cases, and look at how the scientific incentive structure might push us towards performing science that might not be reproducible. I'm then going to focus in particularly and look at replication in animal cognition, behavior, and welfare. So even though a lot of these disciplines are disciplines in animal cognition research, we haven't really had much of a debate about replication so far. Some areas have, but a lot of the case we haven't really looked at what the reliability of our research might be. So I'm going to shine the light onto there. And then I'm going to look in the final section at some methods of increasing reliability and the credibility of our research through what is often called open science practices, things like preregistration, open data, registered reports. But to begin with, what is open science? So you'll hear about it in a lot of different venues. So you'll definitely be familiar with open access, the idea that we should have our publications not behind paywalls, but it also encompasses things like open source software, so things like RStudio, Zenodo, the reference manager, getting education, so open educational resources, and also making a science that's fair and just and open to everybody. So you're going to get a lot of different definitions of open science. I'm going to talk a little bit about open access, and particularly what I think to be ethical open access to begin with, and then I'm going to focus more on the bit which you can't even see under this umbrella of open science, of sort of reliable research. But first we'll have a little detour into open access and the decisions we might face as scientists when we're publishing. So a typical model for how money runs through science is you have a funding body, say the ERC, you'll get a grant or you'll be working on a grant, so you'll be the custodian of that money for a little bit, the public funds, and then you get some data that you want to publish and you think, okay, where do I want to publish this? I want to publish it into a journal, I want my research to be read. And you have a choice between publishers, right? You can choose to go and publish in a nice high impact factor journal. This might be good for your job career. It might actually make your research more visible. But there's lots of different venues you can choose. You could even just choose to preprint something and leave it there and not go any further, and people still might read it. But some of the venues you can choose might be for-profit publishers. So what you might do is pay them a fee, give them an APC, say I'll pay $1,500 or something to publish in an Elsevier journal. And what that does is you're actually giving money to the company Elsevier, which is going to take some away for the profit. So these companies have massive, massive profit margins. And in essence, what we can see from the flow of money is it's going from our public funders through us, and then we've made a choice to give some of that research away from science into the private sphere, into Elsevier's shareholders' pockets. But we do actually face a choice, because we could also publish in journals which are open access and they're not for profit. They should have lower APCs if they're not taking a profit margin off. And there's some open access journals, even in animal behavior, cognition, welfare, that charge 0 APC for fully open access publishing. And of course, preprints are fully open access and free as well. And this is retaining the money in the science. And I think this is something that we should all consider when we're publishing things, because a lot of the times we might perceive it to be better for ourselves to go to the highest impact factor journal possible, or one with a big name. But we have to be careful that we're not just, that we're using our grant money and the public funds as well as possible. And I think also oftentimes the ethical journals, what I call ethical journals, going to be promoting the best science, the most rigorous science. They're not going to be looking for you to put a glamorous headline out there. They want genuinely good quality science, which I'm not sure that journals that for-profit publishers are so keen on all of the time. But we can talk about this later. I just wanted to throw that one out there to begin with as a little token to open access science. So we want open access. I think we can all agree with that. We also want ethical open access. Okay, so briefly moving on now to the replication crisis. So we might have heard of this. It's not so prevalent in animal behavior, cognition, welfare research yet. I think it might be coming up sooner, but it has been very, very prominent across psychology and biomedical science for a long time. A little caveat here, because I want to provide a sort of broad introduction. We're not going to go into a lot of specifics, and replication research is very, very complicated. It's not super clear whether how robust we would want findings to be, whether all true findings should replicate and things like that. But we're going to go at a very surface level as a sort of introduction. So why might research struggle to replicate? And the first thing we want to do is look at the incentives that researchers are under, the pressures that we are when we're generating data and generating research. And we find now that there's a bit of a split between what we would think it would be to be a good scientist and what we would think you need to do to be a successful academic. So a good scientist is, or according to the norms, is very skeptical of the results. They're not going to go and tell people they've found something before they found it. But to be a successful academic, maybe what you want to do is you want to push your paper, you want to oversell it a little bit, you want to get it into a journal which you think is going to get you exposure, you want to get talks, and all that sort of stuff. So maybe there's an incentive pushing you towards claiming things that you not quite have the data to claim yet. Very, very similar. Interpret your that's the same point. I'm sorry, I don't know why that's in there. To be a good scientist, you want to publish your negative results. If you want to be a successful academic, you might want to publish strategically. I feel this barrier is being worn away slightly at the moment. But you hear a lot of times of cases of people want to sell a certain narrative with their research. They only want to publish things which agree with their theories. You have preferred theories. So this might be something, again, which is a conflict between being a good scientist and a good academic. Being a good scientist means not fishing around in your data to try and push things over significance thresholds. Successful academic, okay, well, if you're contingent on making a claim, it means you have to have a p-value of less than 0.5. You're going to fish around in your data. You're going to find lower p-values. And this can be very easy to stumble across lower p-values and over-exaggerate your evidence. And yeah, again, these other things, not paying APCs to get into target journals versus publishing what I perceive to be ethically, there's always this disconnect between, unfortunately, in the incentive structure between what is a good scientist and what is a successful academic. But times are changing. So now we're going to move on to an argument, kind of a famous argument now about why most published research findings are false. Ioannidis made this claim in 2005 that most research findings are false, which is surprising. It surprised me when I first discovered it sort of 4 or 5 years ago. why are we doing it this way if we knew if we knew everything was false in 2005? So I'm going to take you through the Ioannidis argument. So imagine we have a hypothetical research program, and half the hypotheses it tests are true and half are false. So the true ones are out there in green, the false ones are in red. Now we run our studies, and we might detect, say we have 40% power, which animal behavior research, okay, we have noisy animals, we have small sample sizes, we're not going to be detecting things all the time. So let's say we have 40% power for this. Well, we're going to detect 40% of the true hypotheses. For our false hypotheses, we're going to get our 5% false positive rate. So maybe one of these little dots will, by chance, pop out a p less than 0.05. Okay, that would be great if we were doing our analyses with a pre-specified analysis plan. We're going to control our error rate to 5%. But we rarely have this pre-specified analysis plan. We rarely have this control on our false positive results. And what you might see is there might be a little bit of p-hacking, people call it. It's often unintentional, but it's going to be trying a few different analyses out. Oh, that one's significant. Well, maybe we'll roll with that one instead. So the analysis can increase our false positive rate, right? So we see this on the bottom table now, where we now have about 40% false positive, a 40% false positive rate rather than 5%. Okay, that might be a little bit excessive, but we'll go with it. And then on the top panel, some of the true results that we failed to detect, we've now just pushed over the significant result boundary. And this is going to, even though they were originally true hypotheses, we're now going to be overstating the evidence. So these are going to be overestimating the effect sizes that we're seeing. And then the final step, we've run our experiments, we've done our analyses, we're going to go and publish them. But we have a publication bias. Lots of negative results for one reason or other don't get published or haven't been published. So we can now see that what we have on our right is our final literature. And this final literature contains false effects that we can see at the bottom, and it contains true effects that are going to be overestimates of the real effect sizes. And this goes as well for the ones we originally found. So all the ones in green and in light gray. But this might be a relatively typical literature. Researchers under normal pressures might push out that. So when you're studying something which is 50-50 true and false, you might be looking at about less than 50% of your published literature containing true positive results. And you'll be able to see if you change the parameters a bit, if you have more false hypotheses or fewer true hypotheses, lower power, greater power, then you're going to be able to change the numbers that constitute the literature. But you can easily get literatures that are filled with results that are misleading or false, as Ioannidis' argument. And at the end, I've put some links to some critiques of this argument if you're interested in that. Okay, so another piece of evidence we have that a lot of research might be unreliable comes from replication projects. So this is quite a famous image from the Open Science collaboration. And they performed a study in the 2010s where they took 97 original research findings in psychology, so published psychology experiments. And they replicated all of them. They increased the sample sizes a little bit. And what you see here is the results. So on the left, you have the p-values of the original studies. You can see they're all pretty much below this 0.05 threshold for publication. They actually replicated a couple of non-significant effects as well. So they replicate these 97 studies. And then this is the distribution of the p-values of the replication. So you can see now there's a few under 0.05, 37 of them under 0.05, and the rest of them spread out going up to a p-value of 1. So you take 100 studies, replicate them all, and you're getting about a 39% replication rate if you define replication as a significant p-value in the same direction as the original study. So this might be concerning. If we're going into our literatures and we can't replicate the results by doing them again with a slightly higher sample size, this might mean that a lot of our results aren't solid foundations to build on. And this is a one-off study, but now there have been many, many other large-scale replication attempts within psychology, typically within social psychology. And these large-scale replication studies are typically clustering around a successful replication rate of about 60%. This is if you're defining replication success as a significant p-value in the same direction as the original study. And you might say, okay, great, well, social psychology is not animal cognition, animal welfare, animal behavior research, and it isn't. But we are under the same pressures that these researchers are on. We are in the same academic environment, and we also have very similar research methods a lot of the time. Things like using null hypothesis significance testing, small sample sizes, noisy measurements, these types of things. One thing to note is that there's a lot of heterogeneity across replication rates through research methods. So you might find studies which use hundreds of trials on each participant, can get really, really high statistical power, even with small sample sizes. This might be more similar to, I don't know, experiments when you have animals trained on touchscreens or something like that, and they can do thousands and thousands of experiments. You might then be getting quite high statistical power and not churning out so many results that are so unstable. But I still think in these disciplines there are causes for concern, which we'll move on to in a little bit. So why might we be getting these original results that are really significant and then replicating them and not finding the same results again? And one possibility for this is questionable research practices. So experimenters doing, or researchers performing things that might not be exactly what we'd want in an ideal scientific system. And people have done surveys about this. They've gone out, they've emailed authors of journals who've published in journals and said, how often do you do this? How often do you try different analyses before publishing? And you can see here, there's the results from 3 different studies. So 2 studies of psychologists from Italy and the United States, and then a study by Farrar et al. On ecology researchers and evolution researchers. One of the questions they asked was, how often do you exclude non-significant results? How often don't you publish things that don't come out significant? And this is how many people have said they've reported they've done this at least once. And you can see 50%, 60%. Collecting more data after seeing a result's non-significant, and that's going to increase your chances of getting a false positive result, again, around 50% of researchers who performed in these surveys. Rounding p-values, that's one which I find very surprising. So you get a p-value of, say, 0.0505 and then just call it 0.05. A fifth of researchers are saying they've done that at least once. Excluding data points based on significance, so flexibly adjusting the data that they're using in their analyses to see if, presumably to see if they can get a significant result. Again, about a quarter of researchers. So this is quite concerning. If these are relatively normalized research practices, then it might mean that when you open a journal, you see a study, it comes out and it's based on one result and p is 0.03. If you know this type of research is going on, then there's no reason really to believe strongly that that's going to be a stable or true effect. And this can explain why we're getting replication p-values or p-values from replication studies which aren't really that consistent with the original study. So it could be that these original studies are false or misleading or have overestimated effect sizes. It could also be that the effects are genuinely unstable, and we'll come back to that. And changing hypotheses in light of data. So this will give you a false positive rate of 100% if you just make your hypothesis what your data shows you. And again, that's kind of difficult and kind of prevalent. So now to move into animal behavior research. So I've given you some evidence from social psychology, from general research practices, and some surveys, but that doesn't really tell us what's going on in animal behavior. So I've put animal behavior welfare research here in the middle of a few different disciplines. So human behavior science in general, developmental psychology, preclinical animal research, animal learning, and then I'm going to say we're living in the same scientific incentive structure as these fields. And one of the things these fields have done is they've had, in certain areas, pretty rigorous studies looking at the reliability of their results. And everywhere you look, in the related disciplines of animal behavior and animal welfare, we find issues with reliability. We find it in developmental psychology where they use nonverbal tests with infants, just like we do with our animals, looking at eye tracking and things like that. We find it in preclinical animal research where they exclude animals based on or they do this flexibly, look at outliers. If they flexibly exclude outliers, driving significant results forward. We find it in human behavior science, like the open science group that we looked at, and even in animal learning, some of the foundational effects. So the blocking effect. Mace et al. Tried to replicate blocking in animals 15 times and they couldn't get it at all. So we should be very aware in animal behavior and welfare research. There's a large chance that a lot of the effects that we have in our published literatures, especially if they've not been replicated and have quite sort of high p-values, we should be ready for them not to be very robust or replicable. And what this means is, if you're, say you're a student, you're starting your PhD, and you start off and you get a few negative results, you don't know whether it might be that you're unlucky and you just missed the result. You could have had, you could be trying to build on things which just aren't true to begin with, right? You could be trying to build on effects that wouldn't replicate. And that's going to be concerning for animal behavior and welfare research as a whole. So focusing a little bit more on animal behavior, there are some reasons why I think it could even be worse than some of these disciplines. So some of our research might struggle to replicate, even more than these fields that we've seen are really, really struggling to replicate their research. We could be concerned with low power, right? We have often very small sizes of sample size. The sample size average for primate research was about 7 from the many primates group estimated that. We know in animal behavior research that there are large experimenter effects. So us as experimenters, we can bias what animals do. This could be biased in coding, or it could actually be biased in when we're interacting with the animals. We all remember the story of Clever Hands, where the horse seemingly was able to do maths, but it was actually just looking at cues that we didn't know we were giving them as, or they didn't know they were giving them as experimenters. So we could be very, very prone to churning out incorrect effects, but this doesn't necessarily mean that they're, or effects that aren't that robust, but this doesn't necessarily mean that they're going to be wrong. We know animal behavior is really, really variable. We know that if you do something at one site with some animals, it might not happen in another site, which is a bit of a concern if we're doing animal research on hard to reach populations. How do you know that your sample of animals aren't just doing something weird that another sample of those animals wouldn't do? So we're going to go into that a little bit more. And then finally, I think we should be very concerned about this because particularly in animal welfare research, our results actually have quite big implications. If we're going to be changing practices, say on farms or things, then we want to know that our research is reliable and robust and generalizable, right? We want to know that what we've done in the lab, if we did it again in the lab, we'd get similar results. And if we do it in a different environment, say on a farm, we're also going to get similar results. So we really need to get this research right if we're going to be informing practice in the real world. So now we're going to talk a little bit about the variability of animal behavior. So Krabatel, in quite a famous study in preclinical research, took 8 strains of genetically identical mice. So they got lots of different samples across 3 different labs. They either got them from breeders or shipped them out to the different labs. They have the identical mice in these labs, and the labs tried to keep them in identical conditions, and they gave them 5 tasks. So this is as similar as you can keep your animals across different labs. And across the 5 tasks, they found that behavior was pretty different in 3 of them. I've shown you here the data from the elevated plus maze, so just how often these mice are entering arms in different arms of the maze in 5 minutes. And you can see from the graph, the x-axis is just different strains of mice, and then the y-axis is the number of times they're going into these arms. You can see that across these different labs, these are genetically identical mice in each set here, are having quite big differences in the amount of times they're moving into the arms of the maze. So we're seeing that when you keep identical animals in identical conditions across different sites, they're behaving differently. And I think this should give us a massive, massive constraint on our expected replicability of animal research. If you can't get identical mice in identical conditions to do the same behavior, what chance do you have of getting a chimpanzee in a zoo to do something exactly the same as a chimpanzee in a lab or something like that, or your animals at one farm get into the behave similarly as animals in a different farm? This might cause a massive constraint on the generalizability of our research. So what can we do to improve the situation? And I think in our behavior at the moment, there's just a big question of, we need to assess the reliability of the research. We have this sort of converging evidence from different fields and our knowledge of the research practices that we might have quite large problems with reproducibility. So one thing to do is just directly test that. Perform replication studies of key effects, or if you're a new student coming into a lab and you want to build a research program, if you're building on anything, you might just want to replicate the previous research, check that it's okay. You can do it directly through replications, but they're quite costly with animals. You can also try to assess it indirectly with meta-research. And I'm going to go through a few questions that you might want to think about whether you can answer them in your own field before moving on to ways where we can improve the reliability of our research. So these could be things through like making better designs. So running studies that would replicate. They might not actually be replicated in the future, but you might want to increase your statistical power. You might want to increase the heterogeneity of your design. These things can improve the reliability of research. We always want to be improving our statistical thinking, but then I'm going to talk about the things which most often come under the open science umbrella. So thinking about ways to increase our transparency, reduce bias, and these are going to come in the form of registered reports and pre-registrations. But first I'm going to drop onto a few meta-research questions that we might have. So I've flashed up on the screen a load of questions. I'm not going to read them all out, but you can read them for yourself if you like. And I want to know, can you answer these questions? In your field, would you be able to say, for the research that I care about, can I answer all of these questions? How often do replications occur? What distribution of statistical power might my research area be achieving? Are these p-value distributions that we're getting consistent with real effects? How often are statistical errors made? What proportion of studies go unpublished? And I don't think until we can answer the majority of these questions, we're in a position to be able to understand the reliability of our research. If you don't know what proportion of studies aren't published, if you don't know the extent of a publication bias, then your published literature, you don't know whether it contains the 10% of experiments where something randomly happened by chance, or whether it's a full description of the research that's going on. And a nice thing for animal research is that we have lots and lots of previous research in psychology to go off. So psychologists have tried to answer all of these questions throughout their replication crisis. There's now relatively established methods, or getting more established methods, to look at all these questions. And I think these are great questions which you can integrate into your research programs if you don't have access to the animals, if you have students that come in that might not be able to test the animals. These are the types of things that we can start asking. Very simple, from just like, how do replication studies occur? Well, you can go take a random sample of journals and just code how many of them are replications. Were they interpreted as successful? And these are the types of things that we want to be able to answer, to be able to assess the credibility of our research. So now I'm going to move on to pre-registrations. So for those of you who haven't come across pre-registration before, it's essentially where you pre-specify your hypotheses and analysis plan before you perform the data collection. So why might you want to do that? Well, we know that these massive problems are people, or error rates can be inflated to upwards of 50% if people try lots of different analyses. So you get your data, you measure sort of 6 or 7 variables, one of them is going to pop out in one form or another as a significant one, and then you could run with that. And this can happen without any sort of negative intention at all. This is kind of just confirmation bias. And if you believe an effect to be there and one of your measures shows it, then you're going to go with it. So this can increase the transparency to others and transparency to yourself. So you know what you hypothesized before you did the study. You know which analysis plan you went for. It also encourages better study design, because you have to think out exactly, okay, well, how am I going to analyze it? What data am I going to collect? And what are my hypotheses going to be done? You can achieve all of this without pre-registration too, but it works. So I'm going to go into an actual pre-registration now. So this is by Fortmeyer's group. And you can see this is publicly available in the Open Science Framework. You can see here, they write down their research questions and the hypotheses that they have. And then if you scroll down it, you can see they have a sampling plan. They tell you any existing data, and they tell you all the way down to the statistical models that they're going to use. So this one was quite a thorough pre-registration, because they go into depth about the analysis plan. I see some pre-registrations where you're just having sort of a couple of sentences about hypotheses. That's good. It's going to mean you can't change your hypotheses after looking at the data, or if you want to do, then it's clear that this wasn't something you predicted. but it won't offer the same protection of inflated error rates as ones where you write down your analysis plan. But I'm actually going to say that pre-registration, if you're going to go for a pre-registration, you may as well go for a registered report. So a registered report is essentially a publication method which splits peer review in two. So you can see that you have a normal sort of study flow in the blue, what shapes are those? Arrows, blue arrows, where you develop your idea, you design your study, you collect your data, you write it, and then you publish it. And you get peer review after you submit your written report. But what registered reports do is they split peer review in half, and you have a stage 1 and a stage 2 peer review. So you submit for peer review after you've designed the study, but before you've collected any data. And this has the benefit there because so you get all the benefits of the pre-registration, you have your analysis plan, you write your whole introduction and your whole methods, and then you get these peer reviewed. So any issues in your methods can be ironed out before you collect your data. You don't have the problem of collecting your data, writing your report, and then getting your reviewers calling back saying, oh, you should have done this. The whole thing's meaningless. You get all that out of the way before you've collected your data. And what happens after stage 1 peer review, if they say, yes, this is good enough, you get an in-principle acceptance. So this is the journal will commit to publishing your study, providing you do it the way you said you were going to do it, independent of the results. So it doesn't matter what results you get, that can get rid of loads of different biases. And it removes publication bias as well, because the journal can't turn around and say, oh, actually, we're not interested in that result anymore. So I think this is a brilliant publication method. It's a no-brainer if you have a study which you're looking to test hypotheses or something. If you know what question you're going to ask at the beginning of the study and it's not super exploratory, then it's perfect, right? You get your peer review, you get input from experts on your design, you collect your data, and then you can just publish it, providing that you don't, there is the stage 2 peer review to check that your conclusions make sense following your data analysis. And I've only heard good experiences through registered reports. And a cool thing is that pre-registration and registered reports seem to work. So this is a bit of a complicated graph, so I'll talk through it quite slowly. This is from clinical trials looking at cardiovascular health. And they mandated pre-registration in the year 2000. So you see the line coming up here, that's when they mandated pre-registration. On the y-axis, you have the risk of the primary outcome. So anything below 1 showed the drug, or I think it was drugs and diets were beneficial. Above 1 is harmful, and the crosses are significant benefits. The red circles are harm, and the light blues are null effects. So you can see before they mandated registration, you got sort of 80% of the drugs were working or claimed to work. You got all these significant results. And then as soon as they mandated public registration, the whole diagram constricts in. All of these drugs or all these trials that were consistently churning out benefits are now turning into null effects. Rather than having 80% benefits, you're having 90% being null effects. Now, correlation doesn't equal causation, but here I think it strongly indicates that this introduction of registration was preventing certain analytical flexibility, people overstating the evidence, and you're getting many more null effects. So to me, this is pretty clear evidence that it works and it stops error inflation, which is nice. So that was just pre-registration. And registered reports, you can find on the left, this was Sheila, I didn't have the reference in there. I'll add it before we make the slides available. Standard reports in psychology, this was from around 2010 to 2015. You were getting 95% of them were supporting the hypothesis that they set out to support. Registered reports where people had to pre-register their hypotheses and analysis plans. So they had this whole stage 1 acceptance, this is what I'm going to do. It's down to 40%. So you see a massive shift in the composition of a literature that's coming from the standard research practices where you don't have to pre-register your hypothesis, you don't have to pre-register your analysis plan, to ones where people have pre-specified what they're going to do. And you see this drop in hypothesis confirmation from like 95% to 45%. And to me, if you're going to believe any of these results, you're going to believe the ones that people pre-registered their outcomes from, right? If you're going to take a drug, you're going to take one of these two drugs here. You're not going to take these ones. Similarly, if you're going to think which of these results is going to be robust, which of the positive results is going to be robust, more likely to be these ones from the registered reports than the standard reports. So this is one really great way, I think, of or showing people that your research is credible. So this is pretty much what I had time for. I didn't want to go, I've gone over my time anyway. There's just so much to talk about. But I think the message that I want to go for is that registered reports and pre-registration are great ways to demonstrate the credibility of your research and to also improve the quality of the research you're going to do. They're not suitable for every format, but I think it's really worth having a go and seeing if you can incorporate some of it. Open science can be difficult, and you kind of just want to go for one step at a time. Maybe it's even just sharing your data to begin with, or doing a brief pre-registration. It can be quite daunting, but I think this is the way to create credible and reliable research, is to go through registered reports. There's lots more that we could have talked about, but I will leave it there. And also, you should definitely consider doing some meta-research. If you can't answer any of these questions, if there's not available in your literature that you work in, then think about going and finding an answer for these questions. They're quite simple studies to perform relative to animal ones if you can't get access to your animals. Or maybe have a look at things like how often the replications studies occur and what the distribution of p-values are. And that is everything from me. On the slides, I've put a lot of extra introduction to open science reading, further reading on everything, things about improving statistics and experimental design. Thank you.
Yeah, thanks, Ben, for this inspirational and simultaneously slightly depressing presentation on the high inclination of questionable research practices, p-hacking, and all similar stuff in fields that are closely related to animal behavior as well. Everybody feel free to drop questions in the chat function, and I will read them aloud to Ben. I have one quick one first, probably one of the most heard when you gave this presentation or a similar presentation, which is with all the often immense pressure that you have on early career researchers, on PhD students, postdocs, people that don't have a permanent position, to publish in high impact journals, to publish, well, significant results, positive results, to publish convincing story. I know that there's no solution, one solution for all of this, how to decrease the pressure and how to get people to, well, not being pushed towards potential questionable research practices. But what are your personal suggestions, like tiny steps that everybody can, in your opinion, take to decrease these incidences and to decrease a little bit the pressure that most people face?
Yeah, so it's tough. And as you say, I think there's no one size fits all solution. I think that a lot of it comes from education and seeking out ways. I mean, personally, I think statistical inference is a key one. A lot of the issues that I've talked about there in terms of reliability come from people not necessarily knowing that some of the things they're doing with their data are going to increase their error rates to a large extent. I know certainly before I got into all of this, when I was starting out, I was doing all sorts of things to my data. I was trying different analyses. I was basically trying doing lots and lots of bad stuff. My error rate would have been through the roof. Fortunately, I only did sort of 1 or 2 studies, and when I was trying things like that, and then I realized through, actually, I think it was Daniel Larkin's one of his Coursera courses. None of the stuff that I touched got published, so don't worry about that. but also it should have been published, so we could say that I did the study and it was a negative result. But I think a lot of the thing comes from seeking out, understanding how you analyze your data, how that influences the reliability of the results. Because I think a lot of people are going to be unintentionally driving up error rates without realizing it. And that will be one key way to stop these results that aren't going to be too reliable going into the literature. Now, that doesn't stop the pressure on researchers, and this is just a massive problem because the pressure comes from all different angles. It's a top-down pressure in many ways. If they're going to be if it's going to be in your requirement to get promoted, then it's very tricky. I think you do kind of, well, at one point you have to take it on yourself that you're not going to do it. But I don't think there's an easy answer. I think there's a lot of inspirational senior researchers, Dorothy Bishop, Chris Chambers in the UK, and Marcus Manafo, who are driving to make this top-down change. And the funders really do recognise that they want reliable research. A lot of this stuff is actually coming from the funding. So I think there's no better time to be saying, no, I don't want to do that. Because I think that in the future, demonstrating a history of open science practice and reliable research practice is actually going to be what's better for you in the future. So I think in some ways it's a false incentive structure at the moment, because I'm confident that in 5-10 years time, people are going to be super impressed. They're going to be super impressed if you've got your code out there and sharing your code, or if you've done a pre-registration or registered report. So I really think that it's now being assimilated into the incentive structure, open science practices. So that's a bit of optimism.
Okay, we have a first question in the chat. The first part of the question is whether these slides and links to the slides will be made available. So I don't know, for you, Ben, you probably have this on some open science framework repository already, but we are aiming to put the PDF slides on such a repository so everybody can check this and get the links from Ben's slides. And the second one is regarding the registered reports. Whether do you usually fill them out through specific journals, or is there a different organization that is dealing with this?
Yeah, so the first one, the slides will be up. I'll put them on my own open science framework page. You'll be able to find it with my name, but also Christian will put it up on the Applying Ethology webinar series stuff as well. So they should be accessible. I can also tweet them. The second part, sorry, I should have mentioned this. Registered reports are only available at some journals. The number of journals offering registered reports is about 260, over 260 now, including several in animal behavior, cognition, welfare research. If I share my screen again, there's a link at the end. If you go to here, the information on registered reports, or this link here, it's the Center for Open Science, and just /RR, that's registered reports, and they show you every single journal that offers registered reports there. Royal Society Open Science is a PLOS as well, so there are some big general journals that will also do it, as well as discipline-specific ones.
Okay, thanks. There's another question from Sarah. She's stating that you showed in the beginning that there's a big gap between good science and being a successful researcher or academic, which makes improvements in the field very difficult. Having dealt, she apparently has dealt with this topic a bit for the past years, and asks what else can be done or what we can do, not only on an individual level, but also as a society, scientific community, to improve or to decrease this gap, or to smaller this gap.
Yeah, that's the $1 million question. I think being well, I think it depends on what stage you're at, right? I think with I do think open science and good research practices are becoming more and more assimilated into the incentive structure, which is great. But at the end of the day, I think, well, our scientific system's embedded also in what our culture is, right? So we have a very quick culture. We want we want to post something, we want to get likes and all that sort of stuff that's going on. And it's science at the moment is happening at such a rapid rate that you're going out, you want to do a study, you want to publish it, get it published, and then get another publication on your list, people to read it, citation counts and things like that. So I think this metricization of science is one of the bigger issues that we need to get rid of. So things like H-index, impact factor, all this sort of consideration. One thing which always puzzles me with science is that how heavily these metrics are used in job applications, right? So other fields have metrics available. Say you're in a school and you're a teacher. The school knows how much your students improve from year on year or something, or what grade your students get. But if you go for an interview to be a school teacher, they don't look at all of your past. They don't demand that you show them the grades that your children got or students got for the last 15 years. They chat to you and see what your attitudes are toward teaching, whether you're a nice person and whether you're going to fit into the team and things. So I think shifting employment from productivity and impact and publication matrices towards just seeing if you're going to get on with the team and be nice and inclusive, and seeing if you have a general commitment to open science, or not open science, but just to shift away from incentives towards funding people who are demonstrating that they want to make science better and fairer, rather than necessarily the most prolific or groundbreaking research, I think is a big one.
Okay, there's still time to put some more questions in the chat box. I have a further one. You were talking about the how to increase robustness and reliability of published results in animal behavior and animal welfare science. I mean, one way would be to increase the power of our studies, and there are several potential ways to do this, basically. What are the ones from your experience, from your read of the literature, that could be, well, relatively easily be implemented and can thus lead to a fast uptake on the reliability of animal behavior and welfare science?
Yeah, so I think there's three main ways to do this. One is trial number. If it doesn't interfere with your experimental design too much, and you can increase the number of trials that your animals do, then go for it. In theory, you can get if you have a hard constraint on sample size but no constraint on trial number, then collect more data, get individual level data. We know animals display individual differences in behavior. You can make it your goal to estimate that. You can see, okay, well, let's try and really get down to predicting what the animals are doing at an individual level. So having this sort of individual grain level of data would be really good. Obviously, that's not possible all the time. Particularly some experimental designs don't even allow for you to do multiple trials on things like that. I'd say the second one is increasing sample size is always going to be good. But if you get a result that's not super clear, then just replicate it. If it's an important finding, then you're going to want to make sure that it's true beforehand. So increasing trial number, replicating. And then the thing which I think is really crucial in our behavior research, and I hinted at it with the mouse example, is to try and get multiple sites wherever possible involved in data collection. We can, and moving to this sort of idea as sites as random factors. So we have, if you're sampling from one site of animals, then that's just, say you have pigs in one laboratory or something, then that's, you've sampled possibly from a normal distribution of laboratory pigs or something, and you don't know whether you've gone from somewhere in the middle or somewhere from the tail ends, right? And so if you're only sampling one laboratory, then you don't know how representative your sample is of the study species that you want to get at overall. So you could find a really robust and true reliable effect in your sample, but it actually has nothing to do with the species in question. My controversial thought at the moment, controversial, I don't know, is somewhere there's a group of animal cognition behavior welfare researchers studying a group of animals that are more representative of a different species than the actual species that they belong to. So you have the overlapping behavior space of different species. If you're sampling from the tail end of one of them, then you might actually be getting data that's more similar to a rhesus macaque when you're actually sampling from Barbary macaques. And that's one reason why I think it's really, really important to, wherever possible, get multiple site data. So yeah, just to summarize, that'll be a trial number, sample size where possible, and replication, and multiple sites.
Okay, thanks a lot. We have 2 more questions on this. Jen is asking, or Jen is mentioning that it's already quite difficult to get funding on doing novel and innovative research. So the assumption is that it would be probably way more difficult to get funding to replicate other people's studies. This is the first part of the question. The second one is whether you can give any advice on how to avoid predatory journals while trying to find valuable open access journals.
Yeah, so the first point is a good one. If you can't get funding to the replication studies, then it's difficult. I think there is becoming more and more funding available for studies looking at the reliability of research. And as I said, funding bodies really, really do care about research. They don't want to think that they're giving money to experimenters who are going to run as many studies as possible, publish as much as possible, but then not all be reliable. So and there are funding bodies, I don't know specifically from lots of different organizations, but I know there are funding bodies which are now really, really shifting towards saying, okay, well, we need to look at the reliability of the data. So actually, I think it can even be a strength in a grant application. But then it goes into the cost-benefit analysis of replication. So maybe there might be some studies that you want to replicate that are going to be quick and easy to replicate. So there's a contrast between, okay, well, what's the importance of replicating it and how much resources am I going to have to put in? And you want to balance those out. If something's really, really important, then maybe you're going to replicate it at whatever the cost is. But then if it's that important, I think you'll be able to make a good case to get funding to replicate it if there are reasons to think that it might not replicate. So I think there's no easy way to say it. There's no easy way to get funding for something, but the funding bodies will, if it's important and to them, then they will fund studies for replications. And on avoiding predatory journals, there's a list of open access journals, Directory of Open Access Journals, I think it's called, or I'm pretty sure it's that. Let me have a look. I'm just going to search it. Yeah, DOAJ. There's a directory of open access journals which are pretty well vetted. I think just we're quite small disciplines in animal behavior, cognition, welfare research. If you want to find a discipline-specific one, then ask around a little bit. Ask on Twitter. Even asking on Twitter is a great way to do it, or ResearchGate or somewhere. So yeah, I think you can ask around people, and yeah, predatory journals aren't great for that reason. I think using your network of researchers that you know and asking around, then you should be able to find out if it's legitimate or not pretty quickly.
Okay, thanks for your answer. There's another one from Pat. She mentioned that one strong concern she hears from colleagues is when she's discussing pre-registrations and open science in general, like preprints or open data sets, that people of fear to be scooped in their research. You mentioned that journals might commit to publish following pre-registration, but is there any way you can prevent that other researchers just look at your pre-registration and publish it somewhere else, or publish your idea somewhere else, and how these concerns can be addressed? And I think probably can also be addressed in terms of dealing and arguing with people who have these concerns. So what's your take on this?
Yeah, so there's the scooping comes up quite a bit in these conversations. You can, with pre-registrations, you can make them private to begin with and put them under an embargo. So you don't have to publicly pre-register. You can pre-register it. It has a timestamp, but it only becomes available, say, a year after you've run the study or even published it. So you can choose when to make it openly available, and you can give reviewers or whatever a private link so they can view it. Yeah, I think scooping, it can be a concern. I've never heard of it happening as a result of pre-registration or registered reports. And from what I've heard, everyone says they have got so many benefits from performing pre-registration or registered reports that there is no fear of scooping from all the people I've spoken to who do pre-registration and registered reports. It doesn't mean it doesn't happen, but I'm sure it happens otherwise. So yeah, there can be a risk if you're publicly stating something before you've even collected the data, but I don't think it's a massive concern. And if it is, you could go for the private pre-registration beforehand. So yeah, that's my take on that.
So just a quick follow-up on this. In addition to this, what is your suggestion for a strategy to convince people who fear this, especially if these people are potential co-authors or your colleagues who are not yet open to these practices because they fear that they might get scooped on the research?
Yeah, I think it's going to depend on who the person is. I would just try and talk to them. If they're set, then they're going to be set. There are ways people have these selfish reasons to do pre-registration and things like that, where you might get increase in citations or things for having people be aware of your work earlier on and stuff like that. I don't think that's the best way to go. I think just talking through the logic of it, asking what their fears are, and then maybe you could sort of do it stepwise. So you could say, okay, well, maybe we'll go for a private pre-registration to begin with. I think just getting people to talk to people who've done registered reports, there's usually only positive things to hear about them. Of course, there's going to be issues, particularly because these are new things. There's going to be problems that crop up. But I don't think there's anything we really can do about people being resistant to it aside from articulating the benefits. That's all.
Thanks. There's one addition from Sarah to your mention of running multiple site studies. Of course, this is basically increasing, or you need to have an increased budget. You need to have more resources and funding to actually run the study. When we look at animal cognition, it might not be possible to have another subject or another group of subjects in a different location that you might possibly test. So she's bringing up that one other issue or one other solution to this might be whether heterogenization of the study population that you have, rather than running simply multi-site studies. Can you put a bit of information on this as well?
Yeah, so I think it's a brilliant thing. Heterogenization is something that we definitely want to be doing within studies. So I think if I go onto my slides here, I have some references at the end. On the penultimate slide, there's 4 references there which talk about improving robustness of research through heterogenizing experimental design. And what we mean by heterogenizing experimental design is, again, so you might view your sample as your sample of animals as coming from a population. You can also view this for the stimuli that you use or the experimenters that are going on. So if you have experimenter bias, for example, or unintentional experimenter bias or experimenter effects, and you're only sampling one experimenter, then that's going to bias your whole research. But if you manage to get sort of 4 or 5 people to go in, then ideally their biases aren't all going to go in the same direction. So you're going to get a more sort of robust estimate or more accurate estimate of the true effect. And this can happen with everything from the stimuli you use, the apparatus you use, the sites that you sample from. So heterogenization within site, if you can't sample from multiple sites, is a great way to increase the robustness of the tests that you do. I would say that within site is not going to address any between site issues, but it's definitely something to do with insight.
Thanks for the answer. There's another question by Pat. Let me just skim through it. She's currently thinking about trying to upload a private pre-registration to the OSF, which then will be time-stamped, so to prevent any temptations or incentives for hacking or p-phishing while not necessarily sharing outside the protocol outside of the group. Okay, this is more, I was thought there's a question at the end. So she's already considering implementing this step with what you mentioned, a timestamp but close, not open pre-registration that you can later on refer to try to get your own incentives under control, basically, or to better control your incentives.
Yeah, I can see in the comments it says that it's not quite a real pre-registration. It definitely is a real pre-registration. Yeah. Absolutely. If it's time-stamped and private, but it says what you're going to do, then you've got all the benefits of pre-registration there, because the benefit is primarily for yourself to begin with, for the validity of your own research, or to be able to check what your hypotheses were and things like that. So that definitely counts as a real pre-registration. And yeah, we should get beyond this thing about something counts as being within open science or not in open science. It doesn't make sense that it's brilliant to go for that. And I would say, I mean, I would be concerned if I was pre-registering something openly for the first time. I'd be like, well, what if I've got something wrong, or what if someone's going to come steal my idea or something? They're valid concerns. If you want to go to begin with private, and then great if you're happy after you've done the studies to open it up publicly, so people can see, okay, well, this is what they pre-registered. Then that's brilliant. It's definitely a pre-registration.
Thanks for your answer. There's no further questions for now, and I think we already we filled 1 hour on this interesting topic. I just would like to thank you again for this incredibly interesting talk, for sharing all this information, for making this information later on publicly. We will have the slides. I will share a link for the slides, and we also will have the recordings of the talk in the channel in case somebody missed the beginning or missed some parts of the talk as well. Again, thank you to you, Ben, for joining us today and presenting this presentation in our webinar. Thanks a lot.
No, thank you. And thanks everyone for coming out and listening.
So next week we will have, on May 11th, we will have Anjali Babar joining our webinar series on how dogs process human emotions and how they are able to catch up on human emotions. So we're looking very much forward to see many of you next week as well. And with this, I say good evening or good day, wherever you are, in whichever time zone you are. And hopefully see you next week or in the following webinars. Bye-bye.